Hi everyone. So as it was said, my name is Amy. I'm one of the fourth year pathology residents at SUNY Upstate in Syracuse. And I'm excited to share with you the ethnic and molecular disparities in prostate adenocarcinoma. So just to start, some background about prostate adenocarcinoma. At least 1 in 8 men will develop prostate cancer, at least in their lifetime, and each man's risk kind of differs based on increasing age or ethnicity, family history, and so you could see at the top left, differentiated by ethnicity, as Dr. Davis had just mentioned, the African Americans have the highest risk of developing, followed by the Caucasians, Hispanics, Native Americans, and Asians. And then on the right-hand side, you can see, again, the prostate cancer mortality rates are highest in African Americans, like more than half than the other ethnicities. And if you notice, Asians have the lowest. And so some of these risks and cancer mortality rates we think are not based on the screening. So if you take a look at the bottom left graph, whether each bar is representing one of the ethnicities and whether they're up to date in screening or not up to date or never been screened, across ethnicities it's pretty equal. So another thing that is known is that there are some differences genetically behind each ethnicity. So, for example, in 2 of these published studies, they found that the ERG gene has alterations common in African and non-Hispanic Caucasian ethnicities compared to the Asian populations. So, as with all cancers, understanding the genetic background will help us to come up with better targeted therapies and improve the patient outcome. So, in order to kind of look into the genetics behind the different ethnicities, we use the CBio portal. And for anyone not familiar with CBio portal, Memorial Sloan Kettering had developed this, and it's kind of like a comprehensive platform in which you can evaluate cancer genomics. So you can look by cancer type, specific genes, specific alterations, and you can compare them across the data that's either published or provisional uploaded to the portal. And even more studies have been added since we had started this project. So we had taken 19 different prostate adenocarcinoma cohort studies of all of the individuals were self-reported ancestry. And you could see on the left-hand side, we had about 3,600 non-Hispanic Caucasians, and this was probably one of our biggest limitations in the study, because we had very little African and Asian self-reported ancestry. And then of the Hispanics and Native Americans, there were too few self-reported ancestry that we weren't able to extend the study to them. But if you could see on the right-hand side, we have some of the other risk factors associated with prostate adenocarcinoma. So age at diagnosis, whether they were minimum age, maximum age, medium age, each of those bars are representing one of the ethnicities, and you could kind of see that age at diagnosis is not really a factor. And then same goes for the smoking status, whether never smoker, former smoker, or current smoker. So this kind of makes us think maybe there's more so these changes across the ethnicities are more so due to the genetics. And we did find, using the CBio portal, that there were either enriched or exclusive genetic variants among each ethnicity. So on the left-hand side, you could see the missense variants that were only identified in the Asian ancestry, as well as 4 genes here that were truncating and copy number variants only seen in Asian ancestry. And then you could see on the right-hand side, we have a variety of copy number alterations, truncating, and missense mutations found only in the African and non-Hispanic Caucasians, but not in the Asian ethnicity. So this kind of suggests that in prostate cancer in the Asian population, maybe copy number alterations are not as frequent. And then among those studies that were included, we had found that the most frequent genes that were altered, FOXA1, TP53, and SPOP, were altered at different frequencies among the different ethnicities. So in blue, you could see that that's FOXA1 was most frequently altered in the Asian population, and then TP53 in orange was most altered in the non-Hispanic Caucasians, closely followed by the Asians. And then for the SPOP, that was kind of altered at a similar frequency throughout the different ethnicities. And then we could see on the bottom graph, represented by the green bar, the genes with truncating mutations that were enriched in the Asians, and these were statistically significant. And if we take all of the individuals from the 19 cohorts and we plot them on a survival outcome curve, we see that in red, that is our non-Hispanic Caucasians with the greatest survival outcome, and then the African Americans in the blue, and then followed by the Asians in the yellow. However, it was not statistically significant. Again, this could be due to the disparity in the number of self-reported ancestry. We looked at the androgen receptor gene copy number alterations, and we found that it was statistically significant alteration in the androgen gene number of frequency of alteration. And so we can see that this might have a consequence to the overall survival. So in that middle graph, the CBio portal doesn't allow you to change colors, but the yellow line in that middle graph is representing the Asian population. And on the right-hand side, the blue line represents the Asian population, but you could see with the androgen receptor gene amplification, overall survival is worse compared to those that are non-androgen receptor amplified. And so we also found that of these genes listed at the bottom on the left-hand side, the orange bar is showing those Asians that had increased frequency, and this was statistically significant. And if we take some of these genes and look at them as co-altered by the androgen receptor gene, for example, with CDX or CDK12, we could see that for both the Asians and the Africans, the overall survival was greatly diminished compared to the non-Hispanic Caucasians. And so this was actually a statistically significant finding. So in summary, the CBio portal is a powerful tool to explore genomic data across multiple studies, and certain genes altered in prostate adenocarcinoma may be exclusive to, enriched among, or occur at different frequencies between the various ethnicities. And so maybe these differences in prostate cancer risk between the ethnicities are due to predispositions or protective factors arising from these genetic differences. And these findings might suggest that prostate adenocarcinoma may be a genetically different disease within separate ethnicities. While not all our findings are statistically significant, we felt this warrants further evaluation. So I just want to say thank you to Dr. Scott Smith for making this possible for me, and thank you for all of you for your attention, and if there's time, I'll take questions.
Thank you very much for that. It would be really nice if you could have an independent data set, right? Completely separate. So I was wondering, I'm going to connect the talk before using the P1000 cohort. Is there any, do you think that could be a useful cohort for you to validate and get access to? And my second question is, are you doing a molecular pathology fellowship after you finish residency? Because you should.
So for the first question, I think, I mean, it definitely could be useful. The CBio portal kind of only has a certain number of studies that were uploaded, so definitely could be helpful. And the second question is, I'm not doing a molecular fellowship, but I guess maybe I should think about it more.
I think you should.
Thank you.
I have a question.
Yeah.
Since you made the connection, I may as well ask a question, but great talk. One of the things I was wondering, I have several actually, but I'm only going to ask you one. Did you, so I think it's fascinating that you found so many missense mutations that are unique to the Asian, group that you were studying. Have you looked at any co-mutation occurrences within those specific mutations? So patients who have multiple mutations, and if there's like an enrichment of any of those?
So we did find I had a slide on the one with the CDK12 and the androgen gene. And yeah, so there with those co-alterations, there was statistically significant worse overall survival in the African and Asian populations. And then we're still kind of looking into some of the other co-alterations and their frequencies.
Okay, great.
We do have a virtual question, but I guess kind of you addressed that. Have you looked at co-mutation frequencies among Asian-specific mutations?
So I don't think that we got a chance to do that, but definitely something to look into.
Thank you.
Thank you.